Let me do that. Perfect.
I
So March will be 2 years. I don't think we'll do one in December because it overlaps with ASH. So I'll be at ASH. If any of y'all are going to be there, send me a message, let me know. I'd love to meet up, have a coffee, have a beer. And then I'm happy to start planning for a monthly in 2022. So please send me an email if you'd like to present. We're just doing usually Diane asked about the updates on the Megakaryocyte Gordon conference. So here, Alessandra, you give the good news.
It will happening. I mean, we are still deciding the dates, but will be at the beginning of March 2023, most probably in Italy. So we voted for Italy, so we want to keep Italy because
we're so excited. Yes. And then I was saying that the hemostasis Gordon conference is on track to happen this summer, the end of July in Waterville Valley in New Hampshire. So that'll be July 2022. And if you're looking for a Gordon conference experience, I will plug this since I'm doing it. I'm organizing a conference in UNC Chapel Hill. It happens every 2 years. It was supposed to happen in 2020. It'll happen in 2022. I can link the information. It'll be great. It'll be short. It's only 2 days, but it's quite small, usually around 200 people. And that will be in April of 2022. So next spring in person. And that you can look out on my Twitter feed or whatever for more information. So we all have so many reasons to keep living and doing science, all these really fun things happening. Oh God, thank you all for being here. I hope you feel as happy as I do. So I don't want to take up any more time. Again, if you want to present at Blend and Bone next year, reach out to me, even if you presented before, since we're going on 2 years now and you might have some new data. And we'll start filling the schedule for 2022. And without further ado, I hand it over to Alessandra.
Thanks. So, perfect. Thank you so much, Kelly. This is really a fantastic initiative you had, and it's amazing how these seminars you organize are keeping us together for almost 2 years. This I'm really happy to be here and to see all of you at least virtually. So today I have a long seminar to do, so I thought that I wanted to take you through a journey on our data on new and old about the role of thrombopoietin on megakaryopoiesis on the basis of this 2 papers that came out. This is the Andras Love and William Van Shanker commentary on the blood paper about the single cell analysis on megakaryocyte. So we suspected that for a while, but now is a reality. We know it. We have different population of megakaryocyte with different function, and they can become they can change orientation megakaryocyte. So the niche producing megakaryocyte, the niche regulating megakaryocyte can become proplatelets and platelets producing megakaryocyte, and then you have the megakaryocyte that regulate immune response and so on. But I told you that we suspected that because if we go back and we analyze all our data and all the new data with this perspective, then we see that megakaryocyte have different function, and they are not only proplatelets producing and platelets producing cells, but the common feature is that at the moment, at least for our results, it seems that thrombopoietin is regulating all the function of megakaryocyte, and this is a particular because you could expect thrombopoietin to regulate just platelet formation and proplatelet formation. But this again is also sort of expected also from all the data produced at Harvard by Joe Italiano, where he demonstrated that thrombopoietin is important for megakaryocyte development and differentiation, but then something is missing. We don't know how proplatelets and platelets are produced, and many groups have worked on that, but we still don't have a final response to this question. So if you start culturing megakaryocyte in vitro, as you can see here, and now we counted that we cultured at least more than 2,000 samples of blood, of human blood, between cord blood, bone marrow, peripheral blood, and so on. And every time is always the same. So in our hands, you don't have to push megakaryocyte proliferation and the stem cell proliferation to have mature megakaryocyte. So for human, in 2 weeks, you have the progenitors that differentiate into megakaryocyte, and they start to increase their diameter. The ploidy is always the 8N are always a small percentage, so you have big megs, but not all are big megs. And even if you prolong the culture, this is still the same. So nothing changes. The percentage of megakaryocyte is high at the end of the culture, as you can see here. And they express all the markers of mature megakaryocyte. So almost 80% of megakaryocyte are mature megakaryocyte, depending on the receptor. They do form proplatelets, as you can see here. They form also human megakaryocyte, form beautiful proplatelets, as you can see here, and they form proplatelets both in suspension and in adhesion to different matrices. But they always, and this is not only our lab, but all the labs that work on human megakaryopoiesis, they have exactly the same result, and this is amazing. So this means thousands and thousands of experiments. You reach with human megakaryocyte the maximum of 15% of proplatelet formation. So 15% of megakaryocyte, between 15 and 20% of megakaryocyte at the end of the culture produce proplatelets and platelets. But only that. No matter what you do, you can add the different growth factor. You can put megakaryocyte in adhesion, but you don't get much more megakaryocyte producing platelets. So the suspect was okay. We are not using the right triggers, but maybe at this point we have different type of megakaryocyte in culture. And we don't know because we have a mixed culture of megakaryocyte. And as you can see here, what is interesting for human megakaryocyte is that if you increase the concentration of thrombopoietin, you don't increase the percentage of proplatelet formation. So it's still the same. You can increase the complexity a little bit, as you can see here. So you have a little bit more branches, but the percentage is always the same. And again, this comes with different blood samples and from many experiments. So this for the type of culture we are doing can be considered a gold standard. Things change if you use a thrombopag. So if you use a TPO-mimetics, things can change a little bit. So we use a thrombopag in our silk model, in our 3D silk model. So maybe also this can be a change that we should consider. And as you can see here, compared to 10 nanogram, but again, I showed you that 100 is doing the same, we have an increase of platelet production with very high concentration and the very high dose of a thrombopag. We don't know exactly what is going on. We are studying other mechanisms of L-thrombopag, not only the impact and its function in activating the MIPO receptor. And all of you have seen the paper that came out last month, I think, from the group of Marta Sola-Wiesner about the capacity of L-thrombopag of being an ion chelator, and specifically an iron chelator, and maybe also Diane Krause can comment on that. So is L-thrombopag, is the data we are seeing depending on the effect on the MIPO receptor and also on other effect from L-thrombopag? We don't know yet. And again, you not only increase proplatelet formation with increasing the dose of L-thrombopag, but you also increase the megakaryocyte output. So you induce megakaryocyte proliferation with L-thrombopag more than with thrombopoietin. But again, you don't induce an increase of maturation because the percentage of the expression of all the markers of megakaryocyte are the same, and the ploidy is exactly the same. So you are inducing proliferation, so you are inducing an increase of proplatelet formation, or the two independently, but you're not increasing the maturation of megakaryocyte. And what is interesting is that if you use thrombopoietin and you add L-thrombopag, so the data I've shown you, it was just comparing thrombopoietin with L-thrombopag only. If you add L-thrombopag to thrombopoietin, like you do in vivo when you treat patient, then you have an additive impact on both output of megakaryocyte and proplatelet formation. So L-thrombopag is doing something more than thrombopoietin. So if you add the thrombopoietin, you have an effect. And this is again in our 3D Silk bio bone marrow. What happens when you treat patient with L-thrombopag, and are we able to mimic the condition in vivo, and to understand? Do we have models by which we can understand what are we doing treating patient with L-thrombopag? So here we use our system, our silk sponge, and now we have a cassette where we can run two experiments in parallel. So we treated the patient and control at the same time in small sponges, and we induced megakaryocyte differentiation and platelet formation directly in the sponge. And as you can see here, we treating both ankyrin, we treat two different inherited thrombocytopenias, and treated both ankyrin-related disease and MYH9 disease. We saw an increase of platelets in our system, but this increase of platelets was pretty variable, as it happened in vivo. And this work was done in parallel with a clinical trial together with Alessandro Pecci. So he was doing the clinical trial. We were treating the blood of patient before, of course, the treatment in a blind experiment in our bioreactor. And the hypothesis was, are we able to mimic the condition in vivo? As you can see here, what we saw is that when a patient responded to the treatment, they were producing beautiful proplatelets only with L-thrombopag. So what I'm missing here is the label. So thrombopoietin is here, and here is L-thrombopag. So what's going on? If you add L-thrombopag to thrombopoietin, megakaryocytes start to produce platelets, while they do not stand any proplatelets in the presence of thrombopoietin only. So what are we doing here? Again, we are stimulating the MIPO receptor from a different angle, or we are doing something else to this patient. When we put the results together from the clinical trial, what we observed is that with our system, we were able to predict the response in vivo with the thrombopag, but only considering the release of platelets. So we are able to count platelets, to collect platelets in a bag, like we usually do with our bioreactor. So we are able to count the platelets, and the platelet count in vitro perfectly correlated with the platelet count in vivo. So the response, the increased platelet count of patient in vitro. And this didn't happen with megakaryocyte output. So I showed you that the thrombopag promote megakaryocyte proliferation, but this is variable and doesn't correlate with patient response in vivo, while platelet release is correlated. So basically, a thrombopag is working on the last phase of platelet differentiation, especially in patient and in these two inherited thrombocytopenia. In order to understand what's going on and the mechanism, we started collaborating with Anna Raslova, and these are just preliminary results that we published, but we are still working on that. And Anna derived iPS cells from different patients, and now we are testing and using her cells, knowing if they derive from responder or non-responder patient, if they are treated, and we are trying to understand what's going on and how we can modulate platelet formation in this disease. As you can see here, the iPS cells mimic the phenotype of the disease, and you see a decrease in platelet formation in the MYH9 related disease. And again, if you put these cells in the bioreactor and you have them differentiating, they form proplatelets. As you can see here, we can collect the release platelet from iPS, and they do respond to a thrombopag. So this was a responder. So we can use this system and as a model to understand the impact of a thrombopag on platelet formation and proplatelet formation from MYH9 related disease, inherited thrombocytopenia. But then going back, what do we do when we culture megakaryocyte in vitro? So when we started culturing megakaryocyte in vitro, we observed that we can have proplatelets, we all do, in suspension, and we can have proplatelets in adhesion. And this, as I told you, happens in 2 weeks for human. And at the end, you have many megakaryocytes. They form beautiful proplatelets, and they can adhere to different matrices in a time-dependent manner. So in the 16 hours, in 16 hours, they form proplatelets in vitro, and in these 16 hours, they do different things when they are in adhesion. So they spread, they rearrange their cytoskeleton, and then they start to detach and form proplatelets. And this is not regulated only by thrombopoietin, but is regulated by the different effect of thrombopoietin on megakaryocyte. And this is dose dependent. So thrombopoietin in a dose dependent manners impact and promote the granulation of megakaryocyte. That it is an important mechanism both in vitro and in vivo for proplatelet formation. And we know that is important also in vivo because, for example, patients with great platelet syndrome, they have a decrease in platelet count. And as you can see here, for delta granules, these are old experiment that I performed with in collaboration with Christian Gachet and Marco Cattaneo, and even older than that. And we demonstrated that the ADP released by megakaryocyte is able to activate some receptors, the cholinergic receptors, and promote megakaryocyte maturation and platelet formation. But how ADP is promoting megakaryocyte function? ADP is promoting megakaryocyte function by regulating calcium signaling. And this is the work that have been carried on by Christian Di Budo in my lab for a long time, and I have many data to show you now about calcium and diseases and so on. So megakaryocyte, they do express the store-operated calcium entry machinery. And this is a complex of proteins present on the ER and on the plasma membrane. And basically, when this complex is activated, the two channels in the ER and in the membranes are activated. So the calcium goes out from the ER to the cytoplasm, and the calcium enters from the membrane to the cytoplasm. What Christian did a few years ago was to understand what is the importance of the entry from the membrane of the calcium and the entry to the cytoplasm of the calcium from the ER, because they have a different impact on megakaryocyte function. And basically, I will not go through all these Western blot, but what happens is that if you chelate the internal calcium with BAPTA, you see a decrease of all the pathways that are implicated in proplatelet formation. So basically, if you block the ER, you don't have proplatelet formation. While if you block the calcium entry from the outside, from the membrane, you block only the interaction of the megakaryocyte with the environment. So the regulation of the megakaryocyte, of the regulation from the environment of the megakaryocyte, as you can see here. So BAPTA is toxic, but if you incubate with a certain concentration of BAPTA for a short time, you can see the effect preserving the viability of the cells. So as you can see here with BAPTA, the megakaryocytes are still, they don't do anything. They don't rearrange the cytoskeleton, they don't produce proplatelets. With no calcium from in the outside, you do have some rearrangement of the cytoskeleton, but in general, you have a decrease of migration, of addition of megakaryocyte to the environment, as you can see here in this slide. So if you block calcium from the outside, you have a decrease in addition of megakaryocyte. So megakaryocyte interact less with the environment. So thrombopoietin is regulating all this, but calcium is regulating different aspects depending on on where it comes from. So putting all together, thrombopoietin is regulating, as I told you, everything. So thrombopoietin is regulating all these features. So it's regulating platelet formation, proliferation, migration, because it's activating the entire complex, not only the ER or the plasma membrane. Then the calcium can do the difference. Is SOCI a specific mechanism, or calcium in general is important to regulate megakaryocyte function? The answer is the calcium in the SOCI seems to be specific. And these are very recent results we just published on membrane hyperpolarization in megakaryocyte. So if you induce the hyperpolarization of the membrane, you see a decrease in platelet formation. And in particular, we study the potassium channel, one potassium channels, and we showed that this potassium channels is controlled by thrombopoietin, as you can see here, and it's on the membrane. And what this potassium channels does is that if it is activated, the potassium goes out from the membrane and the calcium goes in. So you can expect, okay, we have more calcium inside, and so megakaryocytes are more functional and so on. No, they're not more functional. First of all, because calcium is only coming from the inside, and the SOC is not activated, and the ER, it seems not involved. And also this pre-activation of the megakaryocyte led to a decrease in the stimulation, for example, from ADP or other factors. So megakaryocyte, at the end, they release less platelets. So it's not, calcium is not important in general, as I told you, but it is important a certain mechanism that lead to calcium increase in the megakaryocyte. And for proprietary formation, ER is our goal. I mean, we have to study ER and understand what is the impact of ER and its channel in regulating proprietary formation. And we are studying many aspects of the ER now, and it's really interesting, and there are many things that are apparent, are evident to as a regulator of proprietary formation. So going back to our SOCI, as I told you, it depending on the channel you activate, you have different function, and how SOCI works and how these proteins of this machinery work. So this is a publication from last year, and is again from Christian and Vittorio Bonante. And what they demonstrated is that when, and again, is depending on thrombopoietin, when megakaryocytes are not activated by thrombopoietin, they express a complex that is made by one of the protein, the ER protein of the SOCI complex, TIM, calreticulin, and RP57. When cells are stimulated by thrombopoietin, this complex detaches. And so all the SOCI is activated, calcium goes out, calcium goes in, and all the process that I just explained happens. And this leads to a megakaryocyte proliferation if you keep stimulating megakaryocyte, as you can see here, and if you block if you block calcium again from the outside, you have a decrease in proliferation. Of course, the first thing we wanted to analyze was the myeloproliferative neoplasm, so the mutation in calreticulin. And from the work of Stefan Constantinescu in Belgium and William Van Schanker, we know that when calreticulin is mutated, it goes on the membrane, it activates, it binds to the MIPO receptor, and it activates the MIPO receptor. But what happens inside? So is the calcium machinery still active, and this is promoting even more megakaryocyte proliferation and megakaryocyte function? The answer is yes, because especially in calreticulin mutated patient, you see much more megakaryocyte exhibiting a spontaneous calcium oscillation, as you can see here. Of course, the MIPO receptor is active also in JAK2 megakaryocyte, JAK2 mutated megakaryocyte. So you see some activation, some calcium spike, spontaneous calcium spikes here, but much more in the calreticulin mutated patient. As expected, calreticulin is on the membrane, is only on the membrane in this patient, only in the calreticulin mutated patient, and the complex with ERP57 and STIM1 is not present anymore. So we don't have the complex, so the SOCI is activated and the calcium is increased. And this leads to an increase of proliferation that even in patient with calreticulin type 1 is decreased by reducing calcium entry and reducing the SOCI machinery. Of course, you cannot target the SOCI machinery to try to decrease megakaryocyte proliferation in MPNs, but knowing this system it's important to go deeply in the mechanism and find maybe the small protein, the small channels we can inhibit affecting just megakaryocyte function. And this is just to summarize this part. So the calcium is important, the autocrine mechanism of regulation of megakaryopoiesis is important, and so calcium signaling regulates megakaryocyte function. Thrombopoietin regulates calcium signaling, sustaining maple activation and all the downstream pathway. And in MPNs, you have a constitutive activation of MPL in one side, but inside, especially in reticulin mutated patient, you have an increase of calcium too. So the activation of thrombopoietin is amplified by calcium, and all the processes deriving from this activation are enhanced, and megakaryocyte can proliferate and produce platelets. But one particular thing of this patient is that despite the fact that they have the receptor activated, they have increased level of thrombopoietin. And this is again a paper that we recently published, and it comes from a collaboration with Karin of Meister. Together with Karin, we had demonstrated that the glycosylation in general is important to regulate megakaryocyte function and stem cell function. And now she has much more data on that. But my group, in collaboration with Karin and Silvia Giannino, demonstrated that this is important for MPNs also. So if you see here, the thrombopoietin levels in the plasma of patient is increased, and this correlates with the allele burden of patient. And as you can see here, you see in these are platelets that platelets in MPN they do express an increase. They have an increased level of glycosylation, as you can see here, and this correlates again with the allele burden. So our hypothesis was that if you have an increase in glycosylation, maybe you have a production of some platelets that are not able to or are more prone to bind to the receptor on the hepatocyte, the actual moral receptor, to promote thrombopoietin synthesis. So basically, the level of thrombopoietin we see are increased because they are not regulated, because the production is stimulated, but some alteration in the glycosylation pathways. And this is the case because together with Karin, we demonstrated with megakaryocyte from MPN, they do express increased level of beta-galT1, that is a transferase for galactose. So what happens is that the normal megakaryocyte, platelets get produced with their sialic acid on their membrane, and only when they are old, they are able to stimulate thrombopoietin synthesis from the liver, while in MPNs, with all the mutation, you have an increase in megakaryocyte of beta-gal, and this leads to the production of platelets that are missing sialic acid. They have more galactose on their surface, and so they may stimulate the production of thrombopoietin from the liver. These are preliminary results we published. We are still working on that, but this can be one of the explanation of the loop that the thrombopoietin is creating in this patient, keeping stimulating megakaryopoiesis and all and megakaryocyte proliferation and so on. And what is interesting is that we also demonstrated that the beta-gal expression is dependent on thrombopoietin, even in healthy control. So as you can see here in MPN, you have an activation of the MPL. So even in the absence of thrombopoietin, you have an increase of beta-gal in MPN. In the healthy control, this is dependent from thrombopoietin, but is the glycosylation pathway is decreased in the presence of ruxolitinib. So it's a JAK-dependent process. So we can modulate this with JAK. But again, we have to understand how and when this process has to be modulated. And going on with thrombopoietin and with MPNs, Alessandro Malara in my lab, you know, this is an old slide we keep showing you, but with also we keep adding new data. So megakaryocytes are able to produce matrices, and megakaryocytes in the bone marrow are immersed in matrices, and these matrices together with thrombopoietin are able to regulate megakaryocyte function and proliferation and platelet formation. So, and especially fibronectin, as you can see here. And the matrix is regulating megakaryocyte function also from a physical perspective. So with its elasticity and stiffness, and this is the work of Vittorio Bonante, and he went back to calcium, and again megakaryocytes express also other calcium channels that are mechanosensors, and they are activated depending on the stiffness of the matrix. And the stiffness of the matrix through these ion channels and through calcium regulate the activation of the beta-1 integrin, regulating megakaryocyte function. So the more elastic is the surface through the mechanosensor, it activates the internalization of the beta-1. So basically beta-1 goes inside, and the megakaryocyte change their interaction with the environment and start to produce platelets because they are not that attached to the environment. So the activation of the beta-1 and the fact that the beta-1 can go inside and outside helps megakaryocyte to have a dynamic interaction with the environment and produce proplatelets. And this is also true in vivo. So if you change the elasticity, and this was done in collaboration with Katia Ravid, if you use an inhibitor of the disease oxidase and you change the stiffness of the bone marrow in vivo, you end up having an increased number of platelets in the peripheral in the peripheral blood, and an increased number of young platelets. So you are producing more platelets if the bone marrow is more elastic in vivo. But megakaryocytes, as I was saying before, they not only interact with the matrix in a TPO and calcium-dependent manner, but they are also able to produce matrices. And again, this is the work that Alessandro Malara started and the first work was published in 2000, another paper in 2011, beside the stiffness one, and is still working on fibronectin. So megakaryocyte express fibronectin. This is dependent on thrombopoietin, and they release fibronectin that helps megakaryocyte interacting with the environment. As you can see here, so megakaryocyte release fibronectin, and fibronectin get assembled around the megakaryocyte depending on the environment, and it mediates the function of the megakaryocyte. And I will show you in a minute that this function is also mediating, the fibronectin is also mediating the third function of megakaryocyte. So now we talked about proplatelet formation, then we started to talk about the niche, and now we can also talk about megakaryocyte and inflammation, all mediated by thrombopoietin and matrices and so on. And then Vittorio Bonante arrived, and he showed that megakaryocytes are able to produce more matrices than fibronectin, and he showed that they are able to produce collagen 3 and collagen 4. And he demonstrated that the production of the matrices by megakaryocyte is dependent on thrombopoietin. So again, thrombopoietin is not regulating only platelet formation, but is regulating also the extension, the release of matrices, so the capacity of megakaryocyte to form the niche and to release matrices. And as you can see here, this time, this feature, this process is concentration dependent. So depending on the process, thrombopoietin can exert its effect in a concentration-dependent manner or independently from its concentration. And this is again in light of this new data that were recently published on the different population of megakaryocytes. We should think about that and about our old result and interpret them with a different in a different light. So as you can see here, again, the matrix is released, and this is dependent from the activation of the pathway. So if you inhibit AKT, you see megakaryocytes do not produce, do not express matrices anymore. This is not a direct effect of thrombopoietin, but this is mediated by TGF-beta. So we all know that megakaryocytes produce TGF-beta, especially in MPNs, but they do produce TGF-beta, release TGF-beta. This is an active TGF-beta. Also in physiology, when they are stimulated by thrombopoietin in vitro during megakaryocyte differentiation, and the thrombopoietin regulates TGF-beta release, and matrix is dependent on the impact of TGF-beta on the stimulation of megakaryocyte by TGF-beta, as you can see here. So if you inhibit the effect of TGF-beta on megakaryocyte, you inhibit the niche, the megakaryocyte function of recreating the niche and creating the matrix niche. And what happens in myelofibrosis? Megakaryocytes are able to produce matrices in myelofibrosis. So the fibrosis in MPNs comes from different cells, but also megakaryocytes. So megakaryocytes, they do produce fibrotic cytokines, of course, but they also produce matrices. Who come first? What is the exact role of matrix in myelofibrosis? The matrix from the megakaryocyte is not known. For sure, is regulated by thrombopoietin, but then we don't know which is the role of this matrix. But you can see here, megakaryocytes, they do produce more matrices stimulated by thrombopoietin, and this is as in the controls dependent from the JAK pathway. So it's dependent from thrombopoietin. If you inhibit JAK, then you inhibit the production of matrices from megakaryocyte. Again, this is dependent from TGF-beta. So the TGF-beta is acting on the stromal cells, on the environmental cells, but also on megakaryocyte. Here is the dosage of TGF-beta we have done in collaboration with the group of Gianni Barosi, and you can see this is the bioactive TGF-beta. There is an increase, of course, in megakaryocyte from PMF, and again the release of matrices is dependent from the activation of TGF-beta. So to conclude this part, is TPO, the thrombopoietin binds to its receptor, then you have a granule release, delta granule, ADP, alpha granules, different molecules, including TGF-beta, and then TGF-beta goes around in the bone marrow, but it binds also to its receptor on the megakaryocyte, and it promotes the synthesis of matrix. And this is after many years of talking and chatting and discussing with Mario Cazzola about the fact that we were sure that megakaryocytes were producing also matrices. He was convinced. So in his review with Robert Kralovics in 2016 in Blood, he mentioned us, and it drew this megakaryocyte. It's as a megakaryocyte in PMF that is a self-producing megakaryocyte. So it produces factors, cytokines, and soluble factors, but also matrix and matrix. Again, what is the role of matrix production by the megakaryocyte in PMF is not known yet. Of course, we are working on that, but for sure, the regulation of thrombopoietin is important. Then you have in fibrosis, you have many matrices and especially, as I told you, fibronectin, and what makes fibronectin special is that what is happening in myelofibrosis is that you don't really have an increase of total fibronectin. Of course, you have a little bit, but you have also a change in the composition of fibronectin. So fibronectin can have these different spliced isoforms and that are not normally present in myelofibrosis in the disease. You have an increase, especially of the EDA isoform, and this is again a work from Alessandro Malara. And what happens is that in general, both megakaryocyte and the environment is producing this matrix that is transforming megakaryocyte in cells that can activate inflammatory response, because EDA binds to toll-like receptor 4, only EDA, not total fibronectin, and this stimulates megakaryocyte proliferation. What is interesting is that the splicing of fibronectin, as you can see here, is induced by TGF-beta. So again, you have few actors in this movie. Thrombopoietin is the main actors. Then you have release factor from megakaryocyte and matrix, and they all play together to induce different process to activate different process in megakaryocyte. So depending on this, on how these factors and these actors combine themselves, you can have a different function in megakaryocyte. And to explain all this, these are all data that I took out again and that explain perfectly what is going on. So if you treat mice, and this is again a work from Alessandro Malara, if you treat mice with chemotherapy in one side and in parallel for one mouse with the chemotherapy, a parallel mouse induced thrombocytopenia with an inhibitor of GP1B alpha. And then you sort megakaryocyte. What you have? You have that when you induce thrombocytopenia, you have megakaryocyte expressing some matrices, especially matrices that are important for megakaryocyte function in terms of platelet formation, and especially collagen 4. When you treat megakaryocyte with chemotherapy, you sort megakaryocyte from the bone marrow, you have totally different megakaryocytes. You have megakaryocytes that express many matrices because they have to rebuild the environment, the niche of the bone marrow. And what is interesting is that when you induce thrombocytopenia, of course, these are all you have very low platelet count, you maintain the other cells and so on. When you induce thrombocytopenia, you don't have a huge increase of megakaryocyte. You have a slight increase of megakaryocyte in the bone marrow after 3 days. They form the platelets and then they go down. On the opposite, when you treat your mice with chemotherapy, you have a huge increase of megakaryocyte. And this, now we know, apparently they are megakaryocyte that form the niche, and not only megakaryocyte that form platelets. These are all together. So you have a mixed population of megakaryocyte that have different function. Finally, just to conclude about the importance of the matrix and about the importance of inflammation also in regulating megakaryocyte function, we started to study some collagenopathy. So this is a mouse model developed by Paolo Bonaldo in Padova, and this is the work of Vittorio Bonante. What Vittorio discovered is that these mice, these are mimicking a severe very rare myopathy that is called Bethlehem myopathy. And both these mouse models and also patients, I will show you in a minute, they have a sort of platelets that are more prone to activation. So our question was, is this a peripheral blood problem? Is this an environment problem because they do not have collagen 6, or is a megakaryocyte problem? So we transplanted this. We did a bone marrow transplant of these mice, and so we transplanted the collagen 6 bone marrow in wild type, and these defects, they were all maintained. So what we showed and we demonstrated that if we treated these mice with rapamycin, we were able to rescue the phenotype and to rescue also all the alteration that these platelets and this megakaryocyte were showing in calcium metabolism, expression of the SOCI machinery, and UPR dysregulation, and so on. And this all is all happening because collagen 6 in both patient and the mouse models is not able to go out from the ER because it has to be assembled. The collagen 6 is made by 3 chains. So if you don't have the 3 chains, it doesn't go out. So the fact that the collagen 6 stay in the ER is increasing, is activating mTOR. So a sort of cascade is activating the UPR. Collagen 6 is regulating autophagy, so autophagy is impaired. At the end, you have a sort of inflammation of the megakaryocyte that produces platelets with a pre-activation state, both as I told you in patient and in mouse models. If you inhibit mTOR, you have a rescue of all this phenotype, and as you can see here, these are patients with they show an increase of the SOCI machinery. They are pre-activated and so on. So again, the fact that thrombopoietin is regulating the expression of matrix by megakaryocyte is important, and is important also in diseases that we didn't expect. Then we are doing a bleeding score of this patient. Again, they are very rare, but we are collaborating with a neurologist in Padova, Elena Pegoraro, and we discovered that sometimes with this patient they bleed, and they bleed because they have pre-activated platelets, and so they don't have platelets that can be activated when is needed. So again, now we have another paper in preparation, and then we are concentrating more on understanding this role of the matrices in megakaryocyte. But again, it's a new aspect that came out looking at the regulation of megakaryocyte synthesis by thrombopoietin. So to conclude this self-produce megakaryocyte matrices by megakaryocyte, we show that TGF-beta can regulate the production of matrix by megakaryocyte. We show that the matrix produced by megakaryocyte together with the environmental matrix is important to regulate megakaryocyte function at different level. And in some diseases in some collagenopathy, we see an alteration of platelet formation because this system is altered both in vivo and in vitro. So to conclude, I have many questions. These are only three because, of course, this new data and looking at all my data with a different approach made me realize that we still have many unanswered questions. And basically, I think we should go back to our data and do everything from scratch. For example, using 3D models. For example, understanding that we have to separate megakaryocyte and so on. So is real that thrombopoietin is regulating all these functions that I showed you. I was working the thrombopoietin concentration and level. Is it important or not? And again, does TPO have an effect also on the regulation of the environment and in general on the bone marrow? And of course, I am more relaxed if I conclude my seminar with some silk. What we are doing is that, of course, we need all the mouse models and we need different models and disease models, but we also need advanced 3D models, I think, because with the silk, we can, in general, with a 3D model of the bone marrow, we can change the elements and understand more precisely what is doing what, who is doing what. And here are some preliminary experiments in the lab, and you can see we are recreating the niche, and these are stromal cells that are producing matrices. Before we were just putting matrix. Now we are recreating the niche with the goal of trying to understand what fibrosis is doing also in a controlled environment such as our niche environment. With this, I thank you, Kelly, a lot. I thank you all for your attention. I thank you all my group, funding of full course, and all the collaborators. Thank you very much for your attention.
Holy shit, Alessandra, that was crazy. An absolute masterclass in megakaryocyte biology. I also feel really jealous of everything that you know. I hope one day I could give a seminar like that. That was amazing. We already have some questions. If y'all have questions, put them in the question and answer or the chat. I'll find them. If you want to stop screen sharing. But also congratulations to all the people listed there. I mean, your people are incredible. Okay, so let's get started because, you know, the people demand more answers. From Anandi, she says, very interesting and great to see you here. Your results overlap with our own findings with the MPN patient platelet transcriptome, identifying ER stress and calcium regulation in proliferation. How do you see some of these findings correlate with platelet thrombotic bleeding phenotypes in JAK2 versus CALR mutated MPNs?
This is one of the main questions. I don't know yet. I don't know if the fact that calcium can be also dysregulated in platelets can affect platelet function. Probably we are not doing that. So we are not studying platelet function in CALR. Many Italians are doing that, so we should collaborate with them. But maybe it can be the case. Maybe the megakaryocyte can release pre-activated platelets if calcium is dysregulated in megakaryocyte. We haven't seen it. We haven't explored that yet.
I just don't even know how. I mean, you do this calcium work so beautifully. And I think a lot of the people here understand, but like it cannot be overstated how hard it is to work with cells. Exactly. So it's just, I'm always in awe of your signaling work. Hervé says, beautiful work. Beta-4 gal-1 knockout have increased integrin beta-1 activation. Do they internalize beta-1 and produce fibronectin normally?
I don't know. We are working on that. I don't know yet. I don't know about the fibronectin, and no, they don't about the beta 1. No, they have a defect of internalization of activation of beta 1. About the fibronectin and about the connection of the two, I don't know yet. This is a very good question.
Yeah, it's good. This is for Diane. Talked about testing iron chelation with TPO compared to Altrombabad, and commented that iron chelation is challenging to titer. Is that something you've thought about?
No, but we are studying that, and I will consider that. This is very important. Yeah, it's important, very important point.
And we have a question here about managing the level of TPO to treat MPM, NPN, or ET. Do you have thoughts about that?
No, we should talk to Karin if she is able to stop in the TPO secretion from the liver. Maybe the one induced by the sialic acid, of course, we can stop all the. But yeah, this can be a good possibility. Yeah.
Yeah. A question from Sumant. Thank you for this awesome talk. Have you checked the role of TRPC in megakaryocyte differentiation?
Not yet.
No.
Not yet. I know that is expressed, but I didn't check the function.
Can I just ask for a preview? What are I know you have those 3 questions at the end. What is the thing that's most interesting to you right now that you're looking at in the next year or 2?
I think the correlation with the environment, I think so. Yeah. Not only the niche in general, endothelial cells, the niche endothelial cells, and what thrombopoietin is doing, because I think this is crucial. And the interaction of megakaryocyte with the niche through the regulation of thrombopoietin. This I think is important, and this is why I want to recreate a niche, because this is.
I agree so much. So I kind of feel like you said, we almost have to redo everything we did before in this new environment. Do you feel like that's kind of valid?
Yes.
Yeah.
Yeah, I totally agree. Because things change so much. I mean, of course, we have basis. I mean, we have our previous results, but then now we have to reinterpret everything from the beginning. This is fun.
Let's start over.
Yeah.
Job security for us all.
Exactly.
All right. Well, this was incredible, right on time. Thank you so much for being here. I'll post this online later. Thank you for sharing. For all of you that are going to be at ASH, I look forward to seeing you in person, or if you're going to be there virtually, and hopefully in 2022, we'll all get to reconnect. So thank you, Alessandra, and I'll see you all soon.
Thank you. Thank you, Kelly, and thank you, everybody. See you soon. Bye.
Bye.
Bye-bye.